Hi everyone, and welcome to the third episode of Morris Animal Foundation's webinar series, Animal News 101, COVID-19 and Coronaviruses at the Intersection of Human and Animal Health. My name is Sean Anderson-Nevi. I'm with the communications team here at the Foundation. Our goal with this series is to cover topics that are of interest to pet owners and animal lovers in general. We get a lot of questions about pet care and animal health here at the Foundation, and so we'd love to share some of our knowledge with you. For those of you who are new to the Foundation, we are a global leader in supporting studies that benefit the health of animals, including cats, dogs, horses, and wildlife. With the help of our generous donors, we fund scientists around the world to do things like develop vaccines, diagnostics, and surgical techniques for serious health issues that animals face. We've been around since 1948, so 72 years now, and were founded by Dr. Mark Morris Sr., a veterinarian and pioneer in animal medicine who recognized a need for research that would improve animal health. Before we get to our presenters, I want to go over some housekeeping items. First, you should see buttons on the right side of your screen for questions, chat, and to raise your hand. If you have a question, please use the question button, but keep in mind we'll save all questions for the end of the webinar to keep things moving. Also, we can't answer specific veterinary questions about your pet. If you have any health concerns, please speak with your family veterinarian directly. We'll be monitoring questions behind the scenes, and if you have a technical question, we'll do our best to help you. We'll have a couple of polls throughout this webinar, and in the interest of time, we'll give everyone about 15 seconds to answer before I close them. We'll also send out a survey about this webinar later, and we'll love it if you would fill it out. Tell us about what you liked and didn't like about it so we can better tailor future webinars to your needs. You can even help us choose future topics. Lastly, this webinar is being recorded, and we'll send out a link so you can watch it again or share it with others. Okay, now I'd like to introduce our Chief Science Officer, Dr. Janet Patterson-Kane, to say a few words. Hi, Janet.
Hello, thank you, Sean, and thanks all of you for joining us today. Since the beginning of this year, the complex story of COVID-19, the disease caused by the SARS coronavirus 2, has involved us all. As this has unfolded, it has emphasised how interlinked we are with animals and our environment, not only in terms of disease passing from animals to humans, but also humans to animals. Additionally, what is happening has illustrated how expertise and work in animal health research benefits everyone, this being an important part of the concept of One Health that unites human and veterinary medicine. In this collaborative webinar, you will hear from 3 different speakers, beginning with Dr. Gregg Dean. He is Professor and Head of Colorado State University's Department of Microbiology, Immunology, and Pathology. Dr. Dean is a Morris Animal Foundation funded researcher who has 2 active grants on feline infectious peritonitis diagnosis, which he will tell you about, and on vaccine development for feline coronavirus. His work brought him to the forefront of efforts to develop vaccines for COVID-19. Dr. Dean is going to speak about his feline work and how he was able to pivot very rapidly to help with this global pandemic. Following Dr. Dean will be Dr. MacDonald Farnham, the Morris Animal Foundation and Dennis and Connie Keller Director of Training for the Global Health Program at the Smithsonian Conservation Biology Institute. Dr. Farnham will discuss their training program and their One Health approach to improving wildlife and disease surveillance in emerging hotspots, using some coronavirus-specific examples. Finally, we will listen to Dr. Klaus-Peter Kepley, a researcher with the Smithsonian Conservation Biology Institute Center for Species Survival. Dr. Kepley will present exciting research on the main receptor for SARS coronavirus 2, which is called angiotensin 1 converting enzyme 2. This receptor is undergoing intensive scrutiny as it is important in understanding how and why the virus infects the species it does. This may allow us to more definitively identify those animal species it infected on the way to infecting humans and assist in the protection of animals, both in native habitats and in human care. Dr. Kepley is also a Morris Animal Foundation funded researcher. We really hope you all find these presentations informative And thanks again for joining us. Back to you, Sean.
Thanks, Janet. And with that, let's introduce our first speaker today, Dr. Gregg Dean from Colorado State University. Dr. Dean, welcome.
Good morning. Can you hear me okay, Sean? There we go. Thank you, Janet, for the invitation to speak today, and thanks, Sean, for handling the technology. I'd like to welcome everyone and thank you for joining us today. As Janet mentioned, I've been funded by Morris Animal Foundation to work on diagnostic tests and a novel vaccine for the feline coronavirus. And today I'll provide a bit of background on that feline coronavirus, a comparison to the new human coronavirus, and then discuss the practice, really the practice of one medicine where research on an animal disease can benefit humans and vice versa. Next slide, Sean. So coronaviruses are fairly ubiquitous. Many species have their own coronaviruses, and coronaviruses have been broken down basically into two clinical forms that either target the respiratory tract, which is shown on the left. There's a pair of lungs down at the bottom, or the intestinal tract, which you can see a picture of the intestinal tract down on the right. And so a couple of species that primarily have coronaviruses that affect the respiratory tract include chickens and humans. And then species that typically have more GI manifestations includes dogs, mice, pigs, cattle, and cats. Another really interesting phenomenon that we recognize certainly in cats, and now we're also recognizing with COVID-19, is that the central nervous system seems to also be an important target, and certainly something that will continue to be of importance as we follow COVID-19. Next slide, Sean. So there are some common features, though, that are shared by coronaviruses across species. And I'll just run through a few of these that are pretty interesting. And these are things that we recognized before the current pandemic, but certainly have held true within this current infection in humans. So number one is that coronaviruses circulate in populations. And they tend to be there because they persist in the environment, and they're highly contagious. So you get this circulation within the target populations. Typically, they are mildly symptomatic or asymptomatic in the young, and that's certainly what we've seen with COVID-19. Generally speaking, treatment has been symptomatic. So if you look across all those species I mentioned before, we have not had really targeted strategies to treat infections. Mostly we treat symptoms. Obviously, we've learned, again, that there is zoonotic potential. We'll hear a little bit more about this later, but of course, we understand that the SARS-2 virus has emerged possibly from a bat that's shown up there in the right-hand corner. And this is another common feature that we see, that bats are able to carry these coronaviruses along with other viruses, and they're very effective vectors for zoonotic transmission to humans. So what's really curious and fascinating is that these viruses rarely cause disease in bats. They carry them, they can transmit them, but they don't become sick from them. Next slide, Sean. So the feline coronavirus is truly a Jekyll and Hyde story. So the most cats around the world are infected during their life by the feline enteric coronavirus. It is endemic, it's worldwide, but it's also typically asymptomatic, maybe causing mild clinical disease that would be characterized by diarrhea, but for the most part, it's asymptomatic. And this is the mild-mannered Dr. Jekyll. However, the enteric virus can undergo mutation in cats to become feline infectious peritonitis virus. There are several clinical forms of this disease, but shown on the right side is a cat with the so-called wet form. You can see that this cat's abdomen is distended with fluid, and there is a sample of that fluid shown in the syringe next to the cat in the photo. This disease is almost always fatal, and this represents the evil Mr. Hyde. Next slide, please. So there are some similarities. There are some observations we've made in cats over the years that have been interesting to see reveal themselves in the human population with this pandemic. Interestingly, when the virus was first sequenced, the human virus, those of us that work on the feline virus immediately saw some distinct similarities and really understood that we were dealing with a virus that probably was going to look a lot like the virus in cats. And so here's some things that we knew about the cat coronavirus situation, and then what we have seen in the human population. First of all, again, there are asymptomatic infections. That is what characterizes the feline enteric coronavirus. And what we have really learned over the last few months is quite prevalent in humans. So that's certainly something we've been hearing a lot about. We have asymptomatic individuals that are shedding virus. How do we test for this? How do we prevent that shed? In cats, we know that we have a situation where they will intermittently shed virus or even persistently shed virus. So there are a number of reports, and we've observed this, where an individual cats without any symptoms at all will shed the virus for years, for months or years. And so this has not been described in humans yet. We're only a few months into this pandemic. So it'll take some time for us to realize whether we have this condition in humans or not. But it's certainly something we need to be looking for. Another important thing that we have known about in cats is that the immunity is not long lasting. So we have been able, over the last few decades in studying feline coronavirus, been able to follow individual animals and understand their immune response and see that indeed it's typically pretty short, on the order of months, less than a couple of years, generally speaking. There have been several publications recently to show that this may also be the case in humans. So this, again, is quite a significant concern in that if people have been infected and recovered, it's possible that they could again become susceptible to infection. And we have seen that in cats. So that's a common part of what we observe in cat populations, is that they can be reinfected. That is still to be determined in humans. Again, this is going to take a little bit of time. But also, what is problematic, and we're all working very hard to change is that we don't have a vaccine. We don't have a vaccine for cats that is effective and safe. And currently, we don't have a vaccine for humans either. Next slide. So I want to talk just a little bit about antiviral drugs. So I mentioned that there has typically not been a specific treatment against the virus for feline infectious peritonitis virus, and that it's typically been symptomatic. However, the last couple of years have really been quite exciting, as a couple of antiviral drugs have been studied and have really shown some efficacy. So on the right-hand side is the citation from a paper published last April, so April of 2019, from Nels Peterson at UC Davis and his colleagues. And this was very exciting to show that there was efficacy and safety of this drug, GS441524, where Nels and his colleagues treated cats and really saw some dramatic recoveries. So on the left-hand side, you can see a cat at the top, very sick. This cat had effusive peritonitis, very ill. Under most circumstances, we would anticipate this cat would die soon. Treated with this drug, this cat recovered, and there he is, looking beautiful, many months later. This drug is remdesivir, which you've heard quite a bit about and has shown significant promise in humans infected with COVID-19. Another drug that Nels and his colleague, Dr. Kim at Kansas State, have also investigated is a different category of drugs. It's a protease inhibitor that's called boceprevir. Again, it has shown some promise in cats and has also been explored as a drug, or a class of drugs at least, to be used in humans. So in both cases, the cat has really identified in severe disease, such as FIP. These two drugs can be effective and have provided some starting point for use of these drugs in humans. Next slide. So why, after all this time, do we not have a vaccine against the feline coronavirus? So the problem, a major problem, is antibody-dependent enhancement. So what is that? On the right-hand side, you can see a cartoon. It's got three parts. Up at the top, there is a virus there that's purple, and it has antibodies, these Y-shaped molecules that are green in this picture stuck on the surface of that virus. And what we hope is that an immune response will stick to the virus and prevent that virus from engaging the host target cells and prevent infection. That's what we'd like to see. But what we have found in cats that happens quite often, especially with different vaccine strategies, is that we have antibody enhancement. And that's shown in the middle and in the bottom panels, where antibody still sticks to the virus, but that antibody now allows that virus to infect a different type of cell. It facilitates the virus entry. So now it is actually helping the virus infect the host and enhancing that infection. And down at the bottom, it's another example of the type of enhancement that can occur where the antibody again sticks to the virus, but then when it engages cells of the immune system, it stimulates those cells to produce more inflammation and cytokines. And so you may have heard about cytokine storms being part of that very severe part of COVID-19 infection. So this is what we've been up against. And this is also a very significant concern with COVID-19. And you can see the title of a paper that was recently published in April, pointing out that we really need to keep our eye on these new vaccines that are into clinical trials now, and be cautious and aware of the potential for antibody-dependent enhancement. Next slide. So the Morris Animal Foundation funded us last year to develop a safe and effective vaccine against the feline coronavirus as a means to prevent that enteric coronavirus from mutating into the infectious peritonitis virus. We focused our approach on small but critical parts of the virus spike protein. We also have included other viral proteins that have not been associated with antibody-dependent enhancement. And we're using Lactobacillus acidophilus, which is a probiotic, as a vaccine vector. So we thought about this. This was our strategy going into these studies supported by MAF last year. We had thought through these challenges. They're the same challenges we're encountering with SARS-2 in people. And so when this virus emerged in people, we were very quickly able to pivot and think about how can we apply this same strategy in people. Next slide, please, Sean. So this is where we're at. We have developed a prototype vaccine. We are testing that right now for its potential use eventually in humans. We were able to engage in this quickly because of the work that we had done funded by Morris Animal Foundation in cats, from all the work that had been done previously by so many outstanding scientists. And what's really interesting, so that the cat and what we know about the cat really is helping us predict what we might see in people. We're taking strategies that we were looking to use to help animals. We're pivoting and we're seeing whether we can use those to help people. But eventually, we're also going to see information, drugs, vaccine strategies, new information emerge from people that will also help inform us in our fight against coronavirus in cats. And just to point out something that's really astounding. So feline coronavirus was identified in 1977. And since then, there's been 743 publications on that topic. Since the SARS-2 epidemic erupted, in this year alone, 2020, there have been 18,353 publications as of 8 o'clock this morning. So my point here is there's a tremendous amount of information being gathered, tremendous amount of money being spent. We can utilize that information so that we can hopefully accelerate our ability to treat and prevent disease in cats as well. Thanks so much.
Thanks, Dr. Dean. Now let's welcome Dr. MacDonald Farnham with the Smithsonian Conservation Biology Institute. Hello, Dr. Farnham. Hey there.
Yeah.
There you are.
Hello. Greetings, everyone, and welcome. My name is Mac, and I'm a public health veterinarian serving as the Morris Animal Foundation Dennis and Connie Keller Director of Training for the Global Health Program at the Smithsonian's Conservation Biology Institute. I'm really excited to be here with you today, and I'd like to kick us off with a short survey. So if you could launch the first survey, please. So, I'll just read it to you, and then I'll keep talking while folks fill it in. So what disease or diseases have been successfully eradicated globally? And while we take a few seconds to fill that in, I just really want to reach out and say thanks again to the Morris Animal Foundation folks for hosting this event today. As we've all been dealing with the COVID pandemic, I think these Animal News 101 webinars have been a great pivot to continue outreach while we go through this. And I'll give you a few more seconds while I do a self-intro, just to say that I have one of these really cool jobs in that I get to work at a zoo and with colleagues and counterparts from all over the world that are dedicated to wildlife and environmental conservation and healthy populations of humans and animals. And in case you get the chance to visit, our office is at the National Zoo in Washington, D.C., which reopens this coming Monday. We're just down the hill from the Elephant Trails exhibit. And well, actually just me, I like to joke that we work in one of the most secure locations in Washington because we're protected by a herd of elephants. And so we got some survey results in, and this is a little bit of a trick question because you're only allowed one answer, and there are two correct answers. So in history so far, there are only two diseases that have been successfully eradicated. One human, which is smallpox, or variole major and minor, and one animal, which is rinderpest. Some of these other examples, like I see a lot of people responded about poliomyelitis or polio, we have pretty effective vaccines for, but even with effective vaccines, a lot of times it's hard to get a successful global eradication due to a number of complicated issues. Okay, if we can switch back to my presentation and go to the next slide, please. So in the next few minutes, I hope to cover a lot of ground. Mine will be a little more global, bigger scale, macro scale than Gregg's and Klaus's after me. I'll share some of our efforts with the Global Health Program and our vested interest in training and capacity building at the interface of humans, animals, and our shared environments. I'll talk a bit about global grand challenges, which include emerging infectious diseases like COVID-19. And I'll talk about why collaborative One Health approaches and understanding are really needed to address emerging diseases at their source. And if we can advance to the next slide, please. So our mission at the National Zoo, under the umbrella of the Smithsonian Conservation Biology Institute, is to save species globally, which really puts all of our work right in the action at this critical intersection between wildlife, domestic animals, humans, and our shared environments. The Global Health Program was established in large part to study population health at these interfaces. And I'll just go real briefly through the photos here, starting clockwise from the upper left. These are some of the folks we work with and train across a broad swath of projects, highlighting field veterinarians, laboratorians, public policy interns, and telemedicine on the last two photos there. Go to the next slide, please. So my work with the Global Health Program focuses on training wildlife veterinarians, One Health, and conservation professionals to bridge scientific knowledge from the Smithsonian to these interfaces where wildlife and humans are in increasing contact. That contact, which unfortunately contributes to habitat loss, disease emergence, and not uncommonly direct conflict between humans and wildlife species. Our Global Health Training Program trains undergraduate, graduate, and postdoctoral levels, both here in the US and around the world. The program utilizes a mix of internships, graduate and postdoctoral fellowships, and international exchanges to build individual knowledge, skills, and competencies for veterinary and One Health professionals. The majority of our trainees conduct focused independent research projects, and we utilize a train-the-trainer approach to maximize our impacts. Next slide, please. So I want to talk about global grand challenges, which include species survival, food insecurity, diminishing natural resources, climate change, loss of biodiversity, and importantly for today's conversation, emerging infectious diseases like COVID-19. These challenges are all highly complex, and we know that all of these challenges are driven in large part by an exponentially expanding global human population, human behaviors, and human activity. What the COVID pandemic is really bringing into sharp focus for us is that today's world is a one world reality. There's no isolation. Wildlife, livestock, people, and diseases do not respect boundaries. We are all interdependent on each other for knowledge, goods, and services across countries and continents. And there's widespread impacts. What happens in one country can quickly spread and does spread to other countries and regions. Such complex challenges require equally complex solutions, and science is showing us that it is key to collaborate across disciplines, cultures, countries, beliefs, and environments to make positive impacts. Next slide, please. So One Health is the name that we put to this realization, namely that human, animal, and environmental health requires collaboration between human doctors, veterinarians, environmental and social sciences, and many more sciences as well, to understand complex challenges and find solutions. Zoonotic diseases are those diseases shared between animals and humans. Usually we think of a zoonotic disease coming from a wildlife host or reservoir and spilling over into domestic animal populations or directly into humans, where they may amplify and quickly spread. However, humans can also transmit diseases to animals, which we've seen just a month or so ago in April with cases of COVID-19 in a Malayan tiger, among other big cats at the Bronx Zoo in New York City. We also have several other examples, including reverse zoonosis of tuberculosis from humans to non-human primates, in particular great apes and elephants. So those are a couple of example reverse zoonoses. We also know that emerging diseases are best addressed as close to their sources as possible before they can emerge into human and domestic animal hosts. So that's why we're working to build wildlife veterinary and disease surveillance capacity with our partners around the globe. Again, looking to connect scientific knowledge and limited resources where the risk of disease emergence is highest. And I hopefully have a couple seconds here just to go through the photos starting on the left here. These photos are all from Koepfli village on Lake Edward on the western border of Uganda. So the border between Uganda and the Democratic Republic of the Congo. So I was there in 2010 working on a large anthrax outbreak in hippos in Buffalo. And the anthrax is naturally occurring disease that pops up occasionally. And of course, a big concern in endangered wildlife is that it's fatal to them, but also that it can affect and infect people that are in contact with those animals. So this outbreak actually ended up being really big. We had about 280 dead hippos, which presents great challenges. We did, of course, have some success there. In this particular outbreak, there were no human fatalities associated with it. But you can just imagine, I mean, here on the left picture, there's fishermen fishing in the lake, and I'm just standing right there on the shore. You can see the hippo on the water there. Right on the shore next to me are marabou storks and a non-native cattle species. And then just a couple hundred meters up the hill, this was later in the day, but there's an elephant running through the village. Next slide, please. In these following couple of slides, I'll take you through some emerging zoonotic disease theory. So the scale on here is time along the horizontal axis at the bottom here, and then a number of cases along the vertical axis or up and down. So we know that from research and science that there are what we call enzootic outbreaks occurring in wildlife species all the time. And that's what they're showing up as green on my screen, hopefully on yours. These bumps along the bottom here are enzootic outbreaks in wildlife. On occasion, there's a spillover event where that disease can spill over into a domestic animal population and then amplify. Also on occasion, these diseases can spill over directly into human populations and again amplify. And you can advance the slide, please. So some of our efforts are really focused on trying to conduct surveillance and determine when and where these enzootic outbreaks are occurring in wildlife before they cause epizootic outbreaks in animals or people. And then really try to focus on geographic areas where we know these things are happening and focus our control efforts there. You can advance the slide, please. So starting from the bottom right here, which is the graphic I was just on, as these diseases often spill over from wildlife into domestic animals and or humans, that's called emergence. So there's many of the factors in disease emergence are listed here, including human encroachment into previous wild areas, translocation of wildlife, climate variability, that goes along with changes in insect vector density and distribution. And then once one of those spillover events occurs, all these other factors contribute to amplification. These are urbanization, population density, and as I mentioned before, increased connections between people and wildlife. And then of course, living in the global reality that we live today, just a plane ride is, by a plane ride, a disease that emerges can get anywhere in the world within less than 24 hours. So these are some of the pandemic risk drivers that we're really concerned about. You can advance the slide, please. So using a One Health approach, we're really trying to get from being reactive, which is on the top of this slide here. So we know these pathogens circulating in animals and diseases emerge. And then we generally in history have only looked into those diseases once it's impacting the human population. So the One Health approach really tries to shift some of this focus upstream, looking for control measures in wildlife and domestic animal populations and ways to prevent disease transmission and prevent emergence. You can advance the slide, please. And this graphic here is trying to show, it's basically a disease emergence risk map. So looking at places where there's increased loss of biodiversity, increased encroachment of human into previously wild areas. This helps us target where we think that our best educated guess is that some of these diseases are most likely to emerge. And those are the areas that we really want to get into and target for disease surveillance. Again, focusing not just on the end result in humans, but on in the wildlife species and populations before they have that chance to emerge. You can advance the slide, please. And this isn't meant to scare everybody, but this is, again, that same risk map, a little updated version, but showing a lot of the diseases that have emerged over the last several decades. We know that an estimated 60 to 70%, 75% of these emerging diseases are comprised of zoonotic diseases. And of these, more than 70% have purportedly originated in wildlife species. So that's a way of saying statistically that we're really worried about diseases emerging from wildlife. A lot of these diseases have been around a long time, things like rabies, tuberculosis, anthrax, and yellow fever. We've had in the last 100 years some more recent entries, including influenza viruses, the emergence of simian immunodeficiency virus and human immunodeficiency virus, West Nile, Zika, and now more recently SARS and MERS and COVID-19. A couple I do want to focus on just for a second are the influenzas and the coronaviruses. Now, both of these are RNA viruses, which makes them more susceptible to rapid mutations and really conflagrates our efforts to develop vaccines for them. In the influenza A virus family, there are 198 different influenza type A subtype combinations, while only 131 of these subtypes have been detected in nature. And as Gregg just touched on, well, on the coronavirus end of things, there are seven types of human coronaviruses currently known, including SARS, MERS, and the recently emerged SARS-CoV-2. So these have been around in human populations for a long time too. And as Gregg mentioned, in animal populations, we look at poultry infectious bronchitis virus, and pigs transmissible gastroenteritis virus, and of course the example of FIP that Gregg just used in cats. Now, we do know that because of sequencing, that the SARS-CoV-2 virus has a stark genetic similarity, about 96% homologous, as we say, so 96% similar with a coronavirus found circulating in bats. And unfortunately, in virology today, even with our most advanced methods, that's about the closest we can get. There's generally not a smoking gun where we can say this virus emerged from this specific species and find that patient zero in the wildlife population. And then just to review that SARS came around in about 2003. About a decade later was the MERS coronavirus. And now in 2019 into 2020, we're dealing with SARS-CoV-2. Go to the next slide, please. So here I'll bring you back to some of our recent work that includes capacity building efforts to conduct disease surveillance in several bat species in caves in Myanmar. My colleagues have just published a study from this work in which they identified 6 new and different coronaviruses from bats sampled in and around these caves. So we know those viruses are there. We know people nearby and in those caves have a relatively high risk of being exposed to those viruses. And together with our partners in Myanmar, we've developed risk communication materials and strategies to better inform and help educate local human populations on the risks of collecting bat guano from those caves, and therefore hopefully preventing disease exposure and potential spillover into humans. This is just one important example of diseases we know are circulating in wild animal populations. We also work with threatened and endangered wildlife species with local partners in Africa, Southeast Asia, Central and South America to not only understand potential disease risks to humans, but to better understand and address the global grand challenges threatening species survival in our shared environments. Advance the slide, please. So this will be my last slide, and I really want to highlight that we're working with great trainees around the world. The photos here are of Dr. Maureen Kamau, who's one of our Smithsonian fellows based at Impala Research Center in Laikipia County, Kenya. Now, Maureen's work, she's a Kenya Wildlife Service affiliated veterinarian, so she gets to go out and help when KWS has wildlife, injured wildlife or problems. She gets to go out and be as a clinical field veterinarian. She works in clinical case management, so helping manage some of those wildlife cases back to health. She's conducting independent research, and the photos here, she's been training wildlife rangers to collect fecal samples from eastern black rhino to track their reproductive status using a non-invasive method. And she's also participating in training students. So down on the lower right here, you can see she's having a couple of veterinary students from the University of Nairobi train on the laboratory techniques in the lab. And then just in broad summary to close this out here, I just want to revisit that we are all connected, humans, animals, and our shared environments. And One Health is that collaborative, multidisciplinary approach we're using to address these complex global health challenges at the interface of humans, animals, and our shared environments. We really think that the best investment for addressing emerging diseases is at the source with local capacity, and the best time to prepare for the next outbreak is now. And I think I might be a little over time, so I might say that we can skip the next survey. Go to the next speaker, please. Thank you guys for for your attention and interest in this.
Thanks, Dr. Farnham. Okay, we'll go ahead and welcome Dr. Klaus Kepley now, also from the Smithsonian Conservation Biology Institute. Let's get his camera up and audio.
Good morning and good afternoon, everyone. And Thank you. My name is Dr. Klaus-Peter Kepley, and I'm a conservation geneticist at the Smithsonian Conservation Biology Institute. So me and my group, we use genomic technologies and data to address questions with regards to endangered species, especially with regards to maximizing genetic diversity. So today, I'm going to be talking to you about our work that myself, as well as a team of 18 other colleagues from Europe and the United States, conducted earlier this year with regards to a particular receptor that is involved in the infection of hosts by SARS-CoV-2. So I'm just going to call it the SARS-2 virus. So next slide, please. So as Gregg and Mac very nicely described already, obviously SARS-CoV-2, this is a zoonotic disease, meaning that it originated from another species or other animals, and we became infected with that virus. And as Mac just nicely told us, is that we're not exactly sure what the origin of this virus was, based on its similarity in terms of the genome of the virus. It does look like it likely came from a bat, but there have been other species that have been suggested as well as potential reservoirs. And again, this is still ongoing research with regards to addressing that question. But in the last few months, as the pandemic has progressed, we've learned quite a lot. Obviously there is human-to-human transmission of this virus, but we've also found examples from different places around the world where this virus can be passed on to cats and ferrets and dogs, and of course the Malayan tiger at the Bronx Zoo. And there are other species as well that become infected with SARS-2 virus, and but just because an animal another species can become infected doesn't necessarily mean that that virus will necessarily replicate. So animals like pigs, chickens, and ducks, these animals can get infected, but it doesn't really seem to do anything to them health-wise. Next slide. So there is a very strong importance of understanding, the susceptibility of SARS-CoV-2 in other species, and there's a number of reasons for this. Where scientists and clinicians are looking for model animal systems that could be used to understand the disease better and its infectivity rate and all the other things that happen when you get sick with this virus. So, and this could be also very instrumental for therapeutic development. Zoos, because of what happened with the Malayan tiger at the Bronx Zoo, and my own experience here at the Smithsonian Conservation Biology Institute and the National Zoo where I'm based, I got involved in the research I'm telling you about today because one of the veterinarians contacted my boss, and my boss contacted me and asked the question do we know anything about whether SARS-CoV-2 is a danger to black-footed ferrets. And black-footed ferrets are an endangered species here in North America that has a very strong 30-year history of captive breeding. And so this got me sort of on this road of looking at these particular receptors that are receptors for SARS-CoV-2 virus in black-footed ferrets and a range of other species. And the questions that we really want to understand with regards to this virus as well as other viruses is what is the range of the species that can serve as hosts? So just because we know that humans can serve as a host, we also want to understand, well, how many other species out there can be susceptible to this virus? And for us, the team of scientists who worked on this project, what we did is we knew we are all genome scientists. We're all part of various international genome consortia where we are working with a team of people around the world to generate high quality reference genomes for vertebrate species around the world. So I'm a member, for example, of the Genome 10K Invertebrate Genomes Project Consortium, which has the eventual goal of generating high quality genome data for all 72,000 known vertebrate species. But we're not there yet. We're far away. And what we wanted to address is that could we use the existing genome data sets, that is the genome data for species that have been sequenced already, to make preliminary predictions about which species are most likely to be susceptible based on receptors that are involved in host SARS-CoV-2 interaction. Next slide. So one of the things that we've understood since the very first SARS virus, and also with SARS-2 virus, is that we know that this virus is very optimized for binding to a particular protein receptor found on cell membranes called Ace2. And Ace2 is just an abbreviation for angiotensin converting enzyme 2. Advance, please.
Yeah, sure thing. Also, Klaus, would you mind turning on your webcam?
Do you need it on or off?
Off. Or I'm saying on.
On. Okay. There we go. Yes. So this ACE2 is found in cells, cell membranes in lungs, arteries, heart, kidney, and intestines. It's involved in regulating blood pressure. Because of that, it's been very well studied with regards to cardiovascular disease in humans. Advance, please. And so we know a lot about this receptor, and it just happened by random chance that this receptor became one of the key receptors that is used by the SARS-2 virus to enter human cells and potentially the cells of other species as well and infect them. And interestingly enough, ACE2, this receptor is found across vertebrates. So we find this in everything from salmon and sharks to frogs and salamanders, lizards and snakes, birds, and all sorts of mammals. So from an evolutionary perspective, we think that this receptor, has had an important function in vertebrate evolution. There's even evidence that some of ACE2 is found in some invertebrate species as well. But given that it's found across vertebrates, we think that this has a very important function. It's been conserved over evolutionary time. And so, next slide. What we wanted to do is that, the spike protein of the SARS-2 virus directly interacts with the ACE2 receptor on the host cells, for example, in humans. And so that's shown there in the picture on the left side there of the slide. And so the SARS-2 virus has these projections that are called spike proteins, and those spike proteins are it's a protein that's consisting of amino acids, and amino acids are the building blocks of all proteins. And a lot of the building blocks, proteins are the building blocks of much of our cells and the functions of our cells and our bodies. And there are particular amino acids of the spike protein that interact with particular amino acids of the ACE2 receptor in the host cells. And so, based on studies that have been done on SARS virus, as well as more recent studies just in the last several months, where because once the pandemic started, the intense scientific interest in SARS-2 virus just exploded. And as Gregg nicely said, he was talking about the number of publications related to cat coronaviruses and so on and so forth, just what's been happening in the last several months. It's the same thing with just the general output of scientific publications with regards to SARS-CoV-2 and its various aspects. And so it is well over more than 50,000 publications that have been coming out and counting. So this is a very intense research topic. And so part of that research has been understanding how the spike protein amino acids interact with key amino acids in the ACE2 receptor. And so, as you see in this photograph, not all amino acids are involved in this interaction. It's only a select few of the ACE2 receptor that are involved in the interaction with the spike protein. And so for our study, what we did is we looked at 25 key amino acids that are involved in binding the SARS-CoV-2 virus across different vertebrates. Advance, please. And so we compared these 25 amino acids in 410 vertebrate species for which there was genome data available on public databases, or data that was generated in each of our laboratories. So for example, my group was working I'm collaborating on, as well as working on a number of genomes that have not been published yet. And so what we did is that we went in and actually mined the ACE2 sequence out of those genomes and then included this into the comparison. And so as you see there from the list of species that we had, it was heavily biased towards mammals because we know mammals are susceptible to this SARS-2 virus. But we also included 69 birds, 17 reptiles, so crocodiles, lizards, and snakes, 4 amphibians, frogs, and 68 fishes. So that's everything from a few sharks to a number of bony fishes. Advance, please. And what we did then is that looking at those 25 amino acids, what we did is we compared those 25 amino acids in those 410 vertebrate species to the human receptor. So we took the 25 amino acids in the human ACE2 receptor as sort of our benchmark, because we know that the human ACE2 receptor is used by SARS-2 virus to infect the cell. And so then we asked the question, well, based on the amino acid differences or similarities in these other vertebrate species, we ranked those species according to very high, high, medium, low, and very low, based on their similarities at these 25 receptors. We also did a number of other studies that confirmed our similarity difference analysis. So on our team, we had a number of structural biologists who basically can model the interactions between particular amino acids in a protein. So our predictions that we set up in this ranking system is that the more different you are at these 25 amino acids, the lower your binding ability would be to the SARS-CoV-2 virus, meaning that what we would predict is that you would have a lower infection by SARS-2 virus. If there are more similarities, then we would predict there is a higher binding ability of the SARS-2 virus and therefore a higher risk of infection. Next slide, please. So just to get into the details of this a little more, on my next slide, this is just the list. That box there shows the list of the amino acids that we focused on. So these are, in science, we usually have a 3 or a 1-letter code for our amino acids. So like that S19 there at the top left-hand corner, that just stands for serine 19. Q24 is glutamine 24, and those numbers refer where they are what their positions are in the amino acid sequence. And the picture on the right side there just shows one of those interactions. So the green structure is just showing a part of the ACE2 receptor in humans, and what I've highlighted there, circled in red, is K31, a particular amino acid that interacts with L455 on the spike protein of the SARS-2 virus. And so this is exactly what we did, is that we looked at these 25 amino acids. We know those amino acids listed there, that's what we see in humans. And then we asked, well, how different are these other vertebrate species with regards to these 25? And then if they are different, does that difference influence the interaction and the binding ability of a particular amino acid at a particular site of the ACE2 receptor? Next slide, please. So what I'm going to show you in the next series of slides is our results. And basically, I've categorized these according to our ranking categories. So these are our very high-risk species. Out of the 410 vertebrate species that we found, these are the ones that are the highest at risk. That is, that those 25 amino acids of the ACE2 receptor are the most similar to the human ACE2 receptor amino acids. And not surprisingly, given their close relationship to humans, all of our species that we found are the old world monkeys and the great apes. So chimpanzees, gorillas, orangutans, drills, gibbons, and snub-nosed monkeys, for example. Those had, at the 25 amino acids, they were exactly identical with the human. And we know from already laboratory studies and some infection studies that old world primates like these animals are at high risk for infection by the SARS-2 virus. The next category are the high risk species. Advance the slide, please. So those included 28 species, and this is where the results really started getting interesting, because we found species that had not identical amino acids at those 25 positions, but near identical. So they just had a few differences, and based on our ranking score and our modeling studies, these would be classified as high-risk species. So these included other old world primates like the lemurs that we had in our data set. But then amazingly, these included animals like whales, like the baleen whales and the dolphins and the porpoises. We had 3 species of deer in our data set, and these 3 species of deer were all very similar in terms of their risk category. We had a number of other rodents like muskrat and Chinese hamster has been shown to be a potential model organism therapeutic model system for studying SARS-2 infection. Next slide. The largest category that we had in our analysis in terms of the higher risk categories are the medium risk species. And so again, this included an interesting array of species. So this includes the New World monkeys, the monkeys that are found in Central America and South America, like howler monkeys, spider monkeys, and capuchin. They also included, this is where we found the cats. Next, advance the slide, please. So we had domestic cat, tiger, cheetahs, and a few other species of cats. All the members of the cat family are found in the medium risk category. And this is, again, consistent with what we understand about this particular group of animals being at risk for infection. Advance the slide, please. The next group was kind of interesting. This is where we found, the relatives of cows and sheep and goats. So these were also ranked as medium risk. So buffalo, sheep, antelope, giraffe are all were included in this category. And then we had a few rodents, not all rodents, but another few distinct group of rodents. And this included the squirrels and the mole rats. So right here I'm showing a marmot and Arctic ground squirrel. These were ranked as medium risk species. Next slide. And then in our lower risk species, this included 49 species from all different groups of mammals. This included like the horses and the zebras, the rhinos, the tapirs, the camels, the dog, members of the dog family, the bears, elephants and manatees and hyraxes were included in this. And this is where we started seeing like the bats falling out. So there were some bats that were included in this low risk category. This means that at those 25 amino acid positions, they were more and more dissimilar compared to the human Ace2 receptor positions. It's also included another group of rodents like the agoutis, for example, were included in the low risk category. And then finally, next slide, the very low risk category. This is where we found the rest of our mammal species and basically all other vertebrate species. So in terms of low risk, this is where we find the seals and the sea lions and the walruses, the ferrets and their relatives like the sea otters. This is where we found the majority of the bats, like the insect-eating bats and the fruit-eating bats, like the fruit bat shown there on the right, the pangolins, and then the rest of the rodents like jerboas and cane rats, for example, were included in this category as well. Advance the slide. And as I mentioned, this is where we found all the other vertebrate species. Oh, please go back one more. So like the sharks and the fishes, the amphibians, the reptiles, and the birds were included in this. They were dramatically different at those 25 positions. And it would suggest, based on our modeling studies, that there would be very, very low chance or low risk of infection by SARS-2 virus of these species because of the difference at those particular amino acid positions. Now you may be asking, well, you've heard that, for example, ferrets can get infected when they are dosed in the laboratory with very high levels of SARS-CoV-2 virus. And again, there were some interesting inconsistencies with our ranking categories and the species that fell into those categories. But again, we are just looking at a single receptor. We know that this SARS-2 virus uses a number of receptors. It could, that is still being worked on, but ACE2 is the highest at the top of the pile in terms of potential candidates for infectivity by the SARS-2 virus. And so in animals like ferrets, we would suggest, based on our studies, that these animals would be potentially infected by SARS-2 viruses using another receptor, another group of receptors. It's the same thing with pangolins. We know that pangolins, and as Max said, bats have a huge diversity of coronaviruses. But again, we think that, as we discussed in the paper that we wrote about our study, that viruses have evolved, bats have evolved with coronaviruses for many, many millions of years. And so this new virus, even though it might have come from a bat, is just something that likely is not going to affect the health of bats in general. And the same thing with pangolins, even though that's still being worked on. So next slide. So just to conclude, I mean, our study is completely was done in a computational way. We took data that was either available from our own labs or available on online databases and did these computational analyses. And it's important to keep in mind these caveats, that our predictions of susceptibility to the virus should be confirmed, of course, with direct experimental data where possible. The pathological outcomes may differ among species, so just because a species may get infected doesn't necessarily mean they will develop some of the symptoms that we find. Because even in humans, for example, there is a spectrum between being asymptomatic and being symptomatic, and even within the symptomatic category, people differ. And this is likely to be very similar in different species as well. And as I mentioned, infection in any species may occur via other cellular receptors, and what we caution in our study is that our prediction shouldn't be overinterpreted, especially when it comes to endangered species and their potential threat. But I think our study provides a useful starting point for the identification of species that may be at risk for human to animal or animal to animal transmissions by this particular virus. Next slide. So I just want to thank my colleagues. It was a wonderful team of 19 researchers from the United States and Europe that got together over nearly daily Zoom calls in April. And literally from the inception of this project to producing a manuscript to submission through a journal was a little more than just 4 weeks. And we basically accomplished this in the month of April while all of us were working at home. And it was just one of the best collaborative experiences I've ever had, because getting the right group of people, and because we're in the particular circumstances and of our interest in our experience with genome data, this was something that we could tackle. And I want to thank all of them greatly. I contributed just a small part of this, and this project was largely led by Dr. Harris Lewin at University of California, Davis, and Eleanor Carlson at the Broad Institute in Cambridge, Massachusetts, and Emma Teeling at the University College Dublin in Ireland. So, and that's it. And I hope you found our results of interest, and thank you very much for your attention.
Thank you, Dr. Kepley. Now I'd like to open it up for questions. And let me open up the cameras for Dr. Dean and Dr. Farnham. If I can't do it manually, if you could both join us, okay? Got one, and then Dr. Dean sent you a request too. So I'll go ahead and start reading questions. If you have questions and you haven't written them already, if you could direct them to one of the specific hosts, that'd be helpful. But I'll start reading with ones that came in shortly a while ago. So question one, how much variation is present in ACE2 in humans, and might variation be a source for the range of symptoms in coronavirus infections?
That's a great question. That is actually something we also addressed in the paper that we submitted. So one of our co-authors from Harris Lewin's lab, Marco Corbo, postdoc, he actually analyzed that. So we looked at the available ACE2 receptors from the 10,000 Human Genome Project, and there isn't that much variation. And for our study, we didn't see a direct correlation between particular variants and susceptibility. That type of analysis is still ongoing right now. So I can't really address that at this point. This is being done at different national levels. For example, there's an Italian study that has been done looking at this question, as well as a Chinese focused study being done. And eventually, I'm sure this will be done for the United States as well. But that's an excellent question, because And again, the preliminary evidence that some studies have suggested there is the particular type of variant you have might make you more susceptible, but it's not conclusive by any means right now. And again, we need to have more control studies to really get at that question.
Okay. Next question. Have you looked at the glycosylation sites near your 25 key amino acids? What do you predict about spike protein binding steric hindrance? Will this change your risk categorizations?
Yeah, also a great question. So this is what some of our structural biologists actually did. And again, I didn't address this, these particular details for this webinar. But yes, so we did look at that for a number of the residues that we investigated, and looked at the rotamer positioning with regards to their binding with the spike protein residues. And what we generally found is that when we found, for example, differences in different vertebrate species with regards to the human and how that influenced the binding category, it didn't really influence the risk of the change in the risk. There were just a few exceptions, and my apologies, I don't recall those right off the top of my head. But in general, our modeling results, in looking at differences from the human receptors, really matched our similarity difference analysis. So the modeling results are largely consistent with what we found. So, but again, this is something we realized, we needed to have. There's just much more room to address these this type of question in much more detail.
Okay. This question might be for Dr. Dean. So, what is the risk of humans transmitting COVID-19 to our house cats? Are pets being tested currently?
Yeah, that's been an important issue, because we do know that cats can be infected, and we know cats can infect other cats. There has not been a documented case of a cat infecting a human so far. There are a few studies that are underway that are trying to generate the numbers. So you can imagine the challenge in doing this sort of study, so that you have to identify individuals that are infected with the virus. Do they have a cat? Test the cat, and then determine which came first. So yeah, the answer is people can indeed infect their cats. And the recommendation is that if someone knows that they've been exposed or knows that they are actually infected, they should avoid contact with their cat.
Okay. Let's go to this one. So, would ACE inhibitors help a prophylactic effect versus COVID-19 infestation?
Boy, I think this is an active area of research as well, and so I don't know if I don't think there is a definitive answer for that right now, but this is being researched.
Okay. Let's go to this one. Is AI being used to review and cross-reference the huge number of scientific studies? Not sure who that's for. Anyone have any thoughts on that one?
I have seen a couple publications along those lines, and I think there is efforts like this that are being done, actually using AI and machine learning to actually look through the literature to provide a synthesis. I actually remember reading a paper about this specifically in April, using machine learning algorithms to actually synthesize all the publications that are now just hundreds every week that are coming out related to SARS-CoV-2 to provide synthesis and even maybe set up like what are the priorities for research directions in different areas related to SARS-CoV-2 research.
Okay. Let's see. If an animal cannot be infected, i.e. get sick, the animal can still be a carrier and shed the virus to others? Any thoughts on that one? Greg? think you turned your microphone off. Self-muted. Hey there? Okay. We'll try to get you back online. We'll go to, oh, there you are. Okay.
So this is an important issue. And to tie this back to everything that Klaus just presented. So one of the questions is, we've had a virus that emerged in China. Maybe it came from bats, it went into people, now it's spread around the world. Can it go into an animal reservoir and reside there to provide continuous exposure and potentially circulation back in the human population? And I noticed that one species that Klaus pointed there was deer mouse. So we know from work that's been done here at Colorado State University that deer mice can be infected and that they can transmit to each other. Can they transmit to people? We don't know. But certainly deer mice are all over the South and Midwest. So there is certainly potential that we could be finding new animal reservoirs for this new human coronavirus that may very well be different and be well informed from Klaus's work, where we should look for these reservoirs as we're thinking about these difficult problems.
Yeah, and this is one of the reasons also why we did our study, is to understand what the breadth of potential animal reservoirs might be based on their risk score rankings based on their Ace2 receptors. So yeah.
How does the virus get transmitted to other species? Is it by consuming the species that carries the virus, or something else?
Yeah, I could take a stab at that one. There are lots of ways. So when we talk about disease transmission, there's direct transmission, there's indirect transmission, or nosocomial transmission. So from example, from a surface that's been touched by an infected person going into another animal. So in wildlife and animal species, I mean, these sort of direct and indirect contacts happen all the time. And it's complex, but what we know about SARS-CoV-2 so far is that it does seem to be spread human to human most easily. It can infect other species, but more difficult and less common. So we basically go with the main tenets of public health, which are wash your hands, cook your food, and don't eat poop. To put it very simply, but again, some of those very basic preventive measures of wearing a mask and social distancing, even with the last couple hundred years of human development of human and wildlife medicine, those are still the most effective means we have to control disease spread. And when we see these things in animal models and other animal diseases, we want to, for example, bovine tuberculosis is infectious to both cattle species and cervids, so deer. So a big portion of the control effort again is to keep deer separate from where cattle are. So again, that physical distancing and face coverings, they're very basic, but they're still the best tools we have to control spread.
Okay. I have heard that blood type makes a difference in humans. Why is that? Or what is that? Any thoughts on that one, guys?
I've heard that as well, but again, that's outside of my area of expertise. So
yeah. Okay. Let's see. I hope this isn't a silly question. I saw dairy cows on your list of species. Could COVID be spread through milk we drink? Klaus, was that your list?
Yeah, I guess that would be my list. Yeah, again, maybe Mac has a better answer for this, but viruses can be, as he just was talking about, viruses can be transmitted in a number of ways, right? So.
Mm-hmm.
Yeah,
What do you
and
think?
I mean, it is a great question, and of course, you just go back to your very basic tenets of public health. Milk is one of the very first public health interventions we dealt with as a country and around the world. I mean, this is the main reason we pasteurize milk and milk products, is to prevent disease transmission. And I'm not confident enough because I haven't actually looked, but certainly high temperature, high throughput pasteurization would kill most viruses and bacterias. So again, we're looking for those, where that risk might be of transmission, just like this question asks, and put control measures in place to manage that risk as best as we're able.
Okay. Well, thanks, Dr. Farnham. I think that will be our last question, just for the purpose of time. So we'll end by thanking our three guests today. Thank you all, Dr. Gregg Dean from Colorado State University, Dr. MacDonald Farnham, and Dr. Klaus Kepley from the Smithsonian Conservation Biology Institute. Thanks for being with us and sharing your insights into the issue of coronavirus. And we want to thank all of you viewers who joined us today. We hope you enjoyed this and found it informative. We'll send a link to this recorded webinar soon if you'd like to watch it again or share it. And we'll hope you join us next month when we'll talk about canine nutrition. So stay tuned for more information about that. Until then, we'll see you next time. Have a great week.
Thanks again.
Thank
Thank you.